Welcome you all. In the last session, you have studied introduction part of microbiology, historical perspectives, important contributions, scope and branches of microbiology. Today in this session, I am going to deal with sterilization. So first we will see what are the learning objectives of this session. So here we are going to study aseptic techniques in microbiology. Aseptic techniques are nothing but sterilization techniques. So, and we can classify the materials which are to be sterilized and which are to be used in the microbiology laboratory. And what are the session outcomes? Actually, we are dealing sterilization definition and what are the principles involved in the sterilization process and what are the advantages of sterilization. I mean, what are the methods which are used to sterilize the different materials which are used in the microbiology laboratory. Next, we will study what is the definition. Actual definition of sterilization is it is a process that destroys or eliminates all forms of microbial life, and it is carried out in healthcare facilities. Okay, healthcare facilities by physical or chemical methods. We can use physical methods or chemical methods to sterilize the articles or objects. So on the whole, we can define the sterilization as eliminate all types of microorganisms. We can eliminate all types of microorganisms in this process, and this elimination can be achieved by various methods like heat, chemicals can be used, radiation methods can be used, and filtration methods are used to do this sterilization process. Actually, these heat, chemicals, radiation and filtration methods we can study in detail in the forthcoming sessions. Some definitions we can discuss here. That is, first one is antiseptics. These these definitions are linked with sterilization process. So we can discuss one by one. Antiseptic is the substance that stops or slows down the growth of microorganism. It will stops or slows down the growth of microorganism, and these are frequently used in hospitals. Usually in microbiology laboratory it is not used, but in hospitals these are used and other medical settings to reduce the risk of infection during surgery and other procedures. So this is the antiseptic. On the whole, we can say it is applied to living objects, that is microorganisms. Second one is disinfection. Disinfection, we can define it as a process that eliminates many or all the pathogenic microorganisms except bacterial spores, because bacterial spores has more harder coat, so that coat cannot be destroyed by using disinfection. So living bacterial spores disinfection process can be used to eliminate almost all the pathogenic microorganisms. And in healthcare settings, objects usually are disinfected. Objects are nothing but materials can be disinfected by liquid chemicals, or it can be called as wet pasteurization. Another definition is bactericidal. It kills the bacteria, so it is called as bactericidal. Next one is bacteriostatic. Here the static means prevention. So the multiplication of the bacteria can be prevented by these type of agents. So these type of agents are used to prevent the multiplication of bacteria. So these are the some of the definitions which you have to study to understand the sterilization process. Next, we will move to applications of sterilization. So what are the applications? It is rapid and thorough. Sterilization process is very rapid, and it is a thorough removal of microorganism can be achieved by sterilization process. And most vegetative mesophilic bacteria can be killed by sterilization process. And this sterilization can be used to sterilize the laboratory glasswares, and surgical instruments can be sterilized. Media which is used in the laboratory, that can be sterilized by sterilization process. And laboratory reagents, different reagents can be sterilized. That is, some of the heat sensitive devices can be sterilized by filtration. That means some of the chemicals are some of the liquids which are heat sensitive. Usually during sterilization process we are giving heat treatment, either by dry heat treatment or moist heat treatment. So when if you are using some of the liquids for the preparation of laboratory culture media and all, that time those liquids, if they are heat sensitive, then those can be sterilized by using filtration method. And sterilization of heat sensitive liquids and gases also, sometimes they are also sterilized by filtration method. And when you compare to hospitals, operation theaters, usually entryways in operation theaters can be sterilized by some of the sterilizing techniques, like UV rays can be exposed to the entryways, and we can remove most of the pathogenic microorganisms, and all types of microorganisms can be uh, eliminate by using this sterilization method. And when you are using the some of the inoculating loops or wires which are used in the microbiology laboratory, those can be sterilized by uh, flaming. And forceps, spatula, which are also used in the microbiology laboratory. And sealed materials like oils, greases, those are also can be sterilized in hot air oven. And rubber items, here we should not use more heat treatment. Okay. And scalpels, blades, glass slides can be sterilized by waving through the flame. And mouths of a culture tubes, bottles, serum, body fluids, these also can be sterilized by using sterilization technique. So these are the some of the sterilization applications or advantages of sterilization, where all we can adopt this sterilization method to sterilize or to maintain the aseptic conditions. Thank you.